Greetings all from Manchester University. I'm now going to spend a few moments to provide you with some insights into my research building flexible biological aerosol detection algorithms for real-time instrumentation. So first, let me briefly explain how a typical UV-LIF instrument works with the WIBS Neo as a primary example. To the right hand side you can see a schematic of the WIBS. The instrument allows for real-time measurements by aerosols by first drawing air in through the inlets before the bioaerosols pass through a 635 nanometer diode laser and are pulsed with xenon flash lamps, which then excite the particular amino acids such as tryptophan or NADPH within the particles, causing them to fluoresce. And it's this fluorescence then which is measured in two wavelength regimes, FL1 and FL2, and over 3 channels. Now these are slightly varied between different instruments. Forward scattering of the particles is also measured using a quadrant photomultiplier tube that allows for measurement of the variation in azimuthal scattering from the particle, and this provides an asymmetry factor. So one of the avenues I'm exploring is developing a robust library of allergenic pollens common to the northwest of England, which you can see pictured, and also just generally to the UK. So using the WIBS then, the idea is to introduce these pollens through isolation experiments and through also chamber experiments using DSTL's Aerosol Challenge Simulator to build a digital profile for each. With this data, I can apply a variety of machine learning methods, either using supervised or unsupervised techniques. A supervised could take the form of sort of a train test split, or a unsupervised could take the form of a hierarchical cluster analysis, such as the one in the top left corner where it distinguished 6 clusters over a period of a couple of months. The endpoint then is to be able to validate and optimize these methods using cross-validation techniques such as silhouette in order to find the optimal clusters, and this can then be applied to a broader ambient data set such as one where we've had the instrument running for a majority of the pollen season to see how well the algorithms can classify or pick up the bioaerosols from that time. This can also then be further enhanced with diurnal plots or MET data such as rainfall to build a bigger comprehensive picture of what is going on. On to my third and final slide then. Another thing we're exploring is the potential for transfer learning and using the quadrant data that comes out of the WIBS in addition to the fluorescence data in some deep learning techniques. So this is where the data is compressed in machine learning to sort of ascertain the important information about the data points. And as the model learns, it's learning features at each layer and then attributes a combination of these features to the specific output. So in other words, the data is compressed. So each time the model learns through a data point, the dimensionality of the image is reduced before it is later increased. And then because the model is required to then reconstruct or decode this compressed data, it has to learn to store all the relevant information and disregard that noise, that background. This encoded or sort of compressed layer, which is represented in the middle there, is also known as the latent vector, which is meaning hidden data. And this is what we're kind of interested in, which can potentially be used to improve the supervised and unsupervised learning techniques for the better classification of bioaerosols. That's a brief overview of my research thus far and my interests. Thank you very much for listening.